Welcome to my class, Dr. Haldar online class. Today we will be discussing on TCA cycle. That is another important cycle we will be seeing today, followed by our previous discussion that is on glycolysis. So what is TCA cycle? TCA cycle is known as tricarboxylic acid cycle, and it was observed that near to 70 percentage of STP, that is adenosine triphosphate, which is the source of the energy in our body's different cells that are synthesized in Krebs cycle itself. And TCA cycle utilized almost two-thirds of the body's required oxygen uh, out of 100 percentage, out of one. The name TCA cycle is used due to the participation of three carboxylic acids, that is citrate, aconitate, and isocitrate. So in our last discussion in glycolysis, we saw that glucose is converted into pyruvate. Here in this cycle, we will be seeing how in TCA cycle pyruvate is utilized and it is ultimately getting converted into carbon dioxide and H2O. So first we'll see how pyruvate is getting utilized in or as a start of the TCA cycle. So here we can see that the end product of glycolysis, that is pyruvate, is getting converted into acetyl CoA in the presence of the enzyme that is called pyruvate dehydrogenase. As I discussed in our last lecture, the what is the role of any dehydrogenase enzyme is it will remove hydrogen from the substrate, and that hydrogen will be accepted by the cofactor known as NAD plus. Once hydrogen will be accepted by the cofactor NAD plus, it will getting converted into NADH. That means NAD will be reduced to NADH in the presence of any dehydrogenase enzyme. And here, in the presence of pyruvate dehydrogenase enzyme, what will happen as NAD plus will be reduced to NADH plus H plus. So subsequently, what will happen? Pyruvate will be oxidized into acetyl CoA. That is the very initial step, or we can say it is the interim step in between glycolysis and TCA cycle. Okay, so the beginning of TCL cycle happens with the conversion of pyruvate into acetyl CoA in the presence of the enzyme pyruvate dehydrogenase, and this enzyme will only act in the presence of five different cofactors named as TPP, that is thiamine pyrophosphate, lipoamide, then FAD, coenzyme A, and NAD+. And here in detail, we can see that when this reaction is happening, when the conversion of pyruvate into acetyl-CoA is happening in the presence of dehydrogenase pyruvate dehydrogenase enzyme, at the same time, what is happening? This carbon dioxide or pyruvate is getting relieved or getting eliminated for the formation of acetyl-CoA. Fine? Okay. Now let us discuss all the steps which are associated in TCA cycle in little detail so that we can understand the reaction mechanism which is involved in different steps of TCA cycle. So this step is over that I told, the interim step between the glycolysis and TCA cycle. Definitely it is a part of a TCA cycle, means if the question will be asked in the exam that write the different steps involved in TCA cycle, so definitely this step has to be the very first and foremost steps. Okay, so we are having acetyl CoA with us as the first step, as the result of the first step of TCA cycle. And then what will happen? The oxaloacetate will come and bind with the acetyl CoA. In the presence of citrate synthesis, it will convert into citrate. Here the interesting thing what is happening, here we can see what is the role of, I mean, here we can see very clearly the role of coenzyme A, the role of coenzyme A in the formation of citrate from oxaloacetate. Why? Because, and how? See here, pyruvate was containing the acetyl group, fine. So the coenzyme A or the cofactor, what it is doing, it is taking away the acetyl group from the pyruvate and getting connected, or it is delivering that acetyl group into the oxaloacetate. So what will happen? Ultimately, it will lead to produce citrate. Once citrate is formed, then in the presence of the enzyme aconitase, what will happen? One water molecule will be relieved. If we see the structure of citrate over here, we can see this hydroxyl group, and here one hydrogen molecule, both will be eliminated as an water molecule. So what will happen? See here the difference between the structure of citrate and cis-aconitate. Here CH2 will be exactly same. Then it will be C double bond O minus, and then here one double bond will come and it will be C double O minus. That means this OH and H molecule from here will be relieved, will be eliminated. So the aconitase enzyme will help to eliminate one water molecule from citrate for the formation of cis aconitate. Now, again, the aconitase enzyme, what it will do in the subsequent step, in the presence of the water, though here the symbol has been given a little, in a little mistakes, we can see here the symbol is showing that the water molecule is getting relieved. However, in the presence of water molecule, what the reaction will happen? Again, one water molecule will come here. So here, hydroxyl group will come, and again here the double bond will break, and hydrogen molecule will come over here. That will lead to form the isocitrate. So what happens? Initially we had pyruvate, then the pyruvate get converted into acetyl CoA. The once acetyl CoA is formed, then along with the oxaloacetate, it leads to form citrate in the presence of citrate synthesis. Then in the presence of aconitase enzyme, ultimately with the relief of one molecule of water and subsequently addition of one molecule of water, it will lead to form the isocitrate. But what the most important thing we can see over here, the CH2C double bond O, which was inherited from the pyruvate, is still been here, and you can see that is highlighted with blue color, fine. And this C double bond O, which is highlighted over here in pink color, is actually the carbon dioxide molecule, or the ionic form of carbon dioxide, which is being inherited from the oxaloacetate. So these are a few very interesting and important steps in structural metabolism, which we need to focus on to understand the mechanism in very detail. Fine. Okay, let's come over here. Our discussion. We had isocitrate to your hand. Now, what will happen in the presence of the enzyme isocitrate dehydrogenase? Again, the dehydrogenase enzyme is coming into picture, and what its role. Now we are knowing that if there is any dehydrogenase enzyme, means that hydrogen molecule will be relieved or will be removed from the substrate, and that will be accepted by some cofactor like NAD plus or something. The same is happening over here. Isocitrate dehydrogenase, what it is doing, it is taking away one hydrogen molecule from isocitrate, and that is being accepted by NAD plus for the conversion into NADH plus H plus. So what is happening? NAD plus is getting reduced into NADH plus H plus, and subsequently isocitrate is getting oxidized into oxalosuccinate. Then again, in the presence of the same enzyme, isocitrate dehydrogenase, what it will happen? One carbon dioxide molecule will be removed or eliminated out from here in this position. So what will happen? This position will be completely vacated now, right? So you see the same structure, only that it is devoid of the carbon dioxide or COO minus in this position, rest remaining in same in the structure, which is known as alpha ketoglutarate. So alpha ketoglutarate will be formed from oxalosuccinate in the presence of isocitrate dehydrogenase. So what is the main thing is happening over here? One carbon dioxide molecule in the form of COO minus will be eliminated out in this step. Fine. So see, we have almost come nearly half of the TCA cycle, and we can see only the participation of the two enzyme over here. One is three enzyme. One is citrate synthase, then aconitase, then isocitrate dehydrogenase. These three enzymes are only acting over here to come up near to the half of the TCA cycle in terms of the production, different metabolic product. Now see the important things what is happening over here. So out of this two carbon dioxide, one carbon dioxide is already relieved or removed. So now and with the structure that is called alpha ketoglutarate. Now again alpha ketoglutarate dehydrogenase will come into the picture. So what will happen? Again the hydrogen molecule will be relieved, that will be taken up by some cofactor. Here again NAD plus is the cofactor, and definitely alpha ketoglutarate will be oxidized into succinyl CoA. Here, the importance of this step, apart from this oxidative reduction, the importance is again another carbon dioxide molecule that was prevailing over here with alpha ketoglutarate will be relieved out. So now this succinyl CoA or this structure of succinyl CoA will be fully devoid of any CO minus, which were being highlighted till this step with the pink color. Fine. So now we are having succinyl CoA with our hand, and the most one of the most important step in TCA cycle is the conversion of succinyl CoA into succinate, because it is the on and alone substrate level phosphorylation in TCA cycle. Like if we can recall, in glycolysis there are two substrate level phosphorylation. If we can recall those steps, one was in the conversion of 3-phosphoglycerate to 2-phosphoglycerate. That was a substrate level phosphorylation step 1 in glycolysis, and the second one is the conversion of phosphoenol pyruvate to pyruvate in the presence of pyruvate kinase. So likewise, here in TCA cycle, we are only having only one substrate level phosphorylation, that is this step, the conversion of succinate from succinyl CoA, and with the presence of the nucleotide that is GTP. GDP will convert it into GTP. At the same time, what will happen? This cofactor COASH will be relieved or eliminated out from succinyl CoA. So what will happen? Only succinate will form. Rest other all will be eliminated out from this substrate or through this step. So now we are having succinate in our hand, and succinate in the presence of succinate dehydrogenase will getting converted into fumarate. So these are the very close and similar structures. And then once fumarate is formed in the presence of the enzyme fumarase, it will get converted into the malate. And finally, malate will be converted into oxaloacetate in the presence of the malate dehydrogenase enzyme. Again, the same thing. Dehydrogenase enzyme is coming into the picture. So what will happen? Hydrogen molecule will be relieved from the malate, and it will be accepted by the cofactor NAD plus, so which will ultimately lead to produce NADH. So it will be a reduction step. At the same time, malate will be oxidized into oxaloacetate. On oxaloacetate will form, then the repetition of the cycle will happen. Means again this acetyl CoA comes into the picture, then again with the help of oxaloacetate, citrate will form. Likewise the cycle will go on, it will continue on. So here we have understood, we try to understand all the metabolic steps along with its mechanism and detailed understanding of the structure, what is happening in different steps involved in TCA. Now we'll be seeing, for all of your understanding, what I did over here is there are multiple steps which are involved in TCA cycle. So what I did, that I have segregated those steps, starting from step 1 to step 10. As we know in TCA cycle, just now we have seen the TCA cycle, there are total 10 number of reactions happening, and what are the respective substrate, what are the enzyme, what are the products, so that it will help us to recall us, it will help us to understand us in more detail. Okay, so just you all can go through once after your understanding over this TCA cycle. And another important thing, most of the competitive exams like GATE, NET, CSI and NET, UGC, NET, even in several university exams, these are the basic question. One of the very basic question I have seen several times, it was asked that whether TCA cycle is aerobic or anaerobic. So these are the questions we frequently get encountered during our different academic examinations, and the answer is very straightforward and clear. Uh, there is actually no direct precipitation or participation of oxygen in Krebs cycle. However, the cycle operates only under aerobic condition. Why so? Because uh, this is due to the fact that NAD plus and FAD from NADH and FADH2 respectively required for the operation of this cycle, which can be regenerated in the ETC cycle only in the presence of oxygen. Therefore, citric acid is strictly aerobic in contrast to the glycolysis, which operates both under aerobic as well as anaerobic condition. So, what is told over here, that TCA cycle is strictly aerobic. Why it's strictly aerobic? Because for the TCS cycle to operate, the participation of NAD plus and FAD, that is the cofactor, is immensely required. And these are the two cofactor which is usually generated during the ETC cycle. We will be knowing what about detail about the ETC cycle in our subsequent lectures. So now for time being, we know we should understand that these are the cofactor which is usually generated in ETC cycle, and that will only be generated in the presence of oxygen. So in that sense, oxygen is strictly required for the generation of NAD plus and FAD in ETC cycle, and in other sense, TCS cycle can only be operated only in the presence of NAD plus FAT. So in that sense, we can say TCA cycle is strictly aerobic in contrast to the glycolysis. As we know, glycolysis can run in both the phases. I mean, in under aerobic condition, under anaerobic condition. By this time, we know the pyruvate is the end product of the aerobic condition of glycolysis, whereas lactase is the end product of the anaerobic condition of the glycolysis. Fine. So these all are the details we have discussed in TCA cycle. Hope this will help all of you to understand uh, the another important metabolic cycle in our biological system. Thank you.